Hello, Arjun.
Okay.
Okay.
Start.
Very good morning to all. Today is the third day of our online lecture series, 1 week online lecture series. And again, welcome to all participants, dignitaries, students, research scholar, and academician in our online lecture series. In third day, the speaker is Dr. Professor Siraj Datta. Sir, deliver this topic and focused this topic on today lecture. The topic is from plant tissue culture to plant genetic engineering. So I would like to request Mr. Arjun Patra, Assistant Professor and Head, Department of Botany, Prabhat Kumar, College Contai. So over to you, Arjun. Please introduce Professor Dr. Siraj Datta in front of our participants. Over to you, Arjun.
Thank you. Good morning to all participants. Again, welcome to Professor Dr. Siraj Datta. Today, the third lecture of 1 week lecture series. Today's speaker, Professor Dr. Siraj Datta, Professor and former head of the Biotechnology Department, Haldia Institute of Technology. Professor Datta has awarded his PhD from Bose Institute and also awarded MTech degree from Indian Institute of Technology, Kharagpur. He has expertise on plant tissue culture, plant biotechnology, plant molecular biology, plant light signaling, and Agrobacterium-mediated gene transfer. Today, Professor Datta will focus on plant tissue culture and also plant genetic engineering, as well as he will focus on the higher education of plant science. Professor Datta has published many research articles on the plant tissue culture in international as well as reputed national journal. 4 students have been awarded PhD from Professor Datta. So welcome to Professor Datta to deliver the lecture in front of our participants. So over to sir. Sir, please.
Good morning, everybody. I hope all of you hear my voice. Voice is clear.
Yes, sir.
Arjun, can you confirm the
Okay,
voice?
sir. Okay,
Yes,
sir.
sir. Yes, continue.
Okay. So today, this is the second lecture, which is delivered by me. We are mainly focusing from plant tissue culture to plant genetic engineering because last day we have discussed why we study the plant, why important of botany, and why we are be proud as a student of botany or student of plant science. And in this connection, I also discussed the plant tissue culture and basic of plant tissue culture, so that today we can focus directly in the plant genetic engineering, because the basic tissue culture or tissue culture is the basic for enter into the plant genetic engineering. So there is some technical error. Excuse me, the slide is not moving.
Sir, wait for some time. It will change.
So you can again share your screen, first off the screen share and again share your screen.
Okay. I hope now it is moving.
Yeah.
Uh, so before entering into the plant genetic engineering, we should first focus on the genetic engineering tool, because we should use some genetic engineering tool to for the for doing the work in plant genetic engineering. So like the our normal instrumental tools, the genetic engineering also having some tools like cutting tool, joining tool, copying tool, then amplifying tool. So these are the tools. So we are like to enter step by step in detail. So before go inside, we are lucidly try to explain all the tools one by one. So first tool is bacteria. Bacteria is one of the tool of plant genetic engineering, because we are using most of the bacteria as a host for the for our plant genetic engineering research or any ordinary technological research. So bacteria is, we all know, it's a prokaryotes, and it's reproduce within the 20. Its doubling time is 20 to 30 minutes. Most of the bacteria, so it's a good host for expressing our protein as well as to multiply our gene. So it's also used to copying our gene. So it's a good host for different purpose of genetic engineering. Then bacterial genome, that are also interesting because it's a single circular chromosome and it won't have any histone protein, and it's very small inside. So we can also sometimes using that bacterial genome to incorporate our gene of interest, and sometimes we are modified the plant bacterial genome for some for some other different purpose. Okay, but basically we focused on plasmid, because mainly the plasmid we are using for cloning our gene or expressing our gene. And plasmid is the unique things of the bacteria. It's not a part of their main chromosome. It's an extra chromosomal circular DNA, and it's not a very useful for main requirement. The main purpose of the nucleic acid is mainly served by the bacterial chromosome, but plasmid have some very specific activity. So we can use this plasmid. We normally use this plasmid for our own genetic engineering purpose, especially the copying the genes and amplifying the genes, or expressing the gene, or getting the desirable proteins. Again, it's hang. Okay, so how plasmid help us? It helps us to insert a new genome into plasmid, which is already said. So we can cut the plasmid, then insert our gene of interest. Kuldeep, please mute yourself. Kuldeep, Geeta, please mute yourself. Then we can produce a recombinant plasmid by using molecular glue. We are discussing one by one. Then we are transforming this plasmid to the bacteria, and we can grow these bacteria for multiply our gene of interest or plasmid, or sometime we are like to express our gene of interest, that means in form of protein. For that we can use the plasmid. So plasmid used to insert a new genome into the bacteria. Suppose the gene we want, that is like insulin genes, lactase, or any hormone, any protein, any small protein, we can identify the gene of interest. Then we can we can cut and inserted our gene of interest inside the plasmid, and then we can incorporate by the ligase, and we can produce a recombinant plasmid. And then recombinant plasmid,
Hello, Dr. Sir.
we are
Hello.
Hello. Then we are focused on the restriction enzyme. Another tools. The restriction enzyme is discovered in early 60s, and initially this restriction enzyme, it's naturally used by the bacteria to protect themselves from the virus. So they are releasing an enzyme which can recognize and cut the viral DNA, so that virus cannot infect the plasmid itself, that bacteria itself. So normally they using this enzyme to protect themselves from the virus. But the beauty of this enzyme is it cannot able to cut the bacterial DNA itself, because bacteria also express an enzyme is called DNA methylase, and that DNA methylase the same sequence which is recognized by the restriction enzyme, so that it cannot cut their own DNA. So your restriction enzyme should recognize which restriction enzyme we are using, normally type 2 restriction enzyme, and that type 2 restriction enzyme normally recognize a very specific sequence. So for that, I'm giving you a clue that rather, madam, I am Adam, then a man, a pan, a canal, a Panama, or was it a bar or a bat I saw. So all the sentences or word we are using here all are palindrome. That means if you read from the font or from the back, we are getting the same word or same sentence. So these are the palindrome. So like that, the restriction enzyme also recognize a palindromic sequence in the DNA, which they recognize and cut in a very specific position. So action of the restriction enzyme. So these are another palindromic sequence, GAATTC. So here GAATTC. So in both the cases, it's a palindrome. So it's recognized by the type 2 restriction enzyme, this particular specific sequence, and all the different restriction enzyme having the different specific sequence, recognizing sequence, and that they are cut either blunt end or sticky end. So here we are just representing the sticky end, not the blunt end, because most of the RDN technology work or genetic engineering work, we are normally prepared the sticky end cutting. Okay. So for the very biggest basic, we are just focusing on the sticky end cutting. So restriction enzyme recognize a specific sequence and cut. So these are their sticky end cutting site. Now this cutting, we can cutting in our gene of interest also. Then we can join together by a molecular group which is called as ligase. So when we join it together, we can express our gene of interest. Like we can isolate a gene from the human, insert in the bacterial genome, and we can transform it to the bacteria, and we can make a human protein inside the bacteria. Now, what are the basic of this expression? The basic of this expression is the codon usage, because we all know the protein having the same code. The bacteria, the code which used for recognize a DNA sequence for their nucleic amino acid, the same code also recognized by the bacteria also. So we can use these things for use the human gene to express in the bacteria, or we can use the plant gene to express in the bacteria, or so on. But I'm not here. The research is going on, and we have some new information that's not included in your undergraduate syllabus, but for your information, some people are not unmute themselves. Please unmute, or else some noise are coming up. So we should, if you have any query, please write in the chat box or in the YouTube chat. I hope the coordinator will inform me accordingly after the end of this session, so that we can smoothly conduct the whole things. So in this connection, I like to inform one thing. That's that there is a new concept. It's not a very new. That's the codon usage. Now we also have after the hello, hello, uh, Dr. Dr. Gautam Ghosh, please unmute yourself, or else some noise is coming up. So in that case, what I like to say, the another information, that is the codon usage or codon preference. Now now the scientists are realize that universal codon table is not it's useful, but not it's 100 fully useful, because some organisms have the some codon preference. For example, like leucine, for leucine we have the 4 different codon for a code. Maybe bacteria are use CEU or CUC, their preference, or plant normally prefer the CUG. So what happened, the if you express the plant gene in the bacterial chromosome, then the most of the codon they are getting, they are CUG, which is not preferred by the bacteria. So their anticodons are not normally present in the their cytosol. So there is a problem to expressing the protein here. So now people are also changing the codon as per the preference of the host, where we like to express the protein. So these things may be a little bit complicated. So I just, for the information, I also inform these things to you for your, it's help you for your higher study. During that time, you may not misconfuse with these things. So now the transformation and the things. So after making the recombinant DNA, we should transform it to the bacteria, and from bacteria, it is expressed. So from DNA to RNA, RNA to protein, protein to the specific trait that we all know. So plasmid, we cut our gene of interest, making a recombinant plasmid, then it transformed to bacteria, then grow the bacteria and harvest and purify our protein, or that is our specific interest. So now we can recapitulate what we learned within this time. What are the different tools? That's the one is cut. So cut. So as you all nowadays during this pandemic situation are more or less fond of with the computer. So we are also accustomed with the language of computer, like cut, copy, and paste. So here the basic molecular biology tool is the cut, copy, and paste. So cut equivalent to your restriction enzyme, because restriction enzyme used for cutting. Then pasting used by the ligase, and the copying by the plasmid. So later on, we also learned the apart from plasmid, we are also use the your PCR or polymerase chain reaction, or for the copying the gene of interest. So these are also used for copying machine or gene xerox machine. People are now it called PCR machine is a gene xerox machine. So I just showing you a small video about
restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Okay, I hope now this part is clear to you. Now the ligase part, ligase can join the DNA with helping to rejoin it phosphodiester bond. Now the another things which we should interesting for as a tool, that's the cloning vector. We already said the plasmid as a cloning vector, but plasmid should have some properties to be a cloning vector, like its small genome size. Its size should be more small, less than 10 kb. Then origin of replication, it should have an origin of replication where we like to grow it, or the host. According to host, your origin of replication should be there so that it can grow well or divide well in the host. Then the selectable marker are normally we are incorporating in that cloning vector or plasmid, so that we can select the transform cell and eliminate the non-transform cell, because selectable marker giving you the opportunity to select the transform cell. And then multiple cloning site, that multiple cloning site is the site where the more than 13 to 14 or more than that restriction sites, recognition sites are present there, so that restriction we can cut and join in that position in our gene of interest easily, and we have some flexibility to choosing our restriction enzyme. Though I present here the very earlier plasmid, that is the PBR T2 picture where the multiple cloning site is not present, but later on the PARC18, PARC19, and so on, all the advanced vector plasmid vector having the multiple cloning site. And also we are now adding another things, that's the reporter gene. That reporter genes also report you that whether the gene of interest are incorporated or it's not incorporated. That things are also report you. So that now we should make our host competent to take up this plasmid, because without making a competent cell, we cannot incorporate our plasmid into the host cell. So for making the competent to the host cell, we are normally treated the host cell to calcium chloride, and that and make and place them in a ice to cool them. And in that time, a calcium are normally attached to the membrane. Calcium ion is attached to the membrane, and when we add the plasmid, and that plasmid DNA making a complex, that calcium phosphate and DNA complex are meeting, and they are adhered, some of them are adhered into the membrane of the outer cell, outer membrane of the host bacteria. And then when we giving a heat shock, that time there some of the plasmids are in taking up taking in by the bacteria or host cell, and by this way we can able to incorporate the plasmid into the host bacteria. And then we are place them in a selectable with grow, we allow to grow with a selectable marker, so that all the transform cells are able to grow, or the bacteria or the host cell which are taking up taking in your plasmid, they are able to grow, and those who are not taking the plasmid, they are unable to grow because of the presence of the restriction, the presence of the your selectable marker or antibiotic resistant marker. So after growing this, we can plate it, and from that plate we can, if you use some selectable reporter gene, then from that reporter gene we can that can report whether your from this plasmid, which plasmid that from the transform plasmid which having your gene of interest and which is not having your gene of interest. So in that case, another thing, another new term is called, it's blue white selection. So in case of blue white selection, though I am putting here the red color, but blue white selection, what happened, the transform plasmid which having your gene of interest are white in color, and those which are not having your gene of interest, they are blue in color. So from that blue white selection, so that are the magic of the your reporter gene, which report you that your gene of interest is incorporated here, that this transform cell having your gene of interest and these transform cells are not having your gene of interest. Now after transformation, we should isolate the our gene of interest if the purpose is copying our gene. So normally for isolating the gene of interest, we are using the different enzymes like lysozyme for the in case of plant cellulase. Sometimes we are using some detergent also, chemical detergent also, to just to burst or to lyse the cell membrane, so that the nucleic acid should come out. And then we are using some protease to remove the protein from the cell, that whole soup. And we are normally using chilled ethanol and salt for precipitate the salting out the DNA, and we can then collect the DNA. Then another tools is come into the picture. Then the DNA which we isolated, how we can visualize it. Normally we are using a tool, it is called gel electrophoresis. Normally with the basic principle that DNA having the negatively charged, and if you use a matrix, then using that matrix, if you in two pole, if you use one negative charge and positive charge, DNA migrate from the negative to positive. So by this way, we can migrate the DNA, and according their size, they are normally differentiated from the lower, that small fragments are go faster, and the bigger fragments are go slower. So by this way, we can visualize the DNA if you use the. So to visualize you, I have a small video.
Doing gel
You can
electrophoresis.
watch it.
The general electrophoresis equipment includes a power supply, gel mold and form, and gel box. Here we have the gel mold, form, and two combs. Carefully take the mold and place it into the form, and place your two combs in. Choose the combs according to how many samples you have. I have a total of 10 samples, so I will have two 8-well combs. For these particular samples, I prepared a 1% agarose with ethidium bromide, 1x TAE buffer. When working with chemicals like ethidium bromide, ensure you're wearing proper personal safety equipment. As you pour the gel, ensure you do not form any bubbles. Also, when you are done, check the bottom of the combs just to ensure you also do not have bubbles there. When your gel turns opaque, it will be ready to use, as you can see here. Next, we must prepare our samples for loading onto the gel. I have a total of 25 microliter reactions. Therefore, I will be using 2.5 microliters of a 10x loading dye. This loading dye will contain 2 dyes so I can monitor electrophoresis, and also glycerol to help weigh down the samples into the wells as I load them. To prevent contamination, ensure you use new tips between each sample. Repeat these steps until all your samples are prepared for loading. Now you're ready to load your gel into the gel box. Carefully remove your gel from the form and place into the gel box. Keep in mind your DNA will migrate to the positive pole due to the negative charge in the phosphates in the DNA. Today I'll be using 1x TAE buffer. Carefully pour the buffer into the gel box, filling both chambers. All you need is a thin layer covering the top of the gel. When this is done, carefully and slowly remove the cones from the gel as to not tear the gel accidentally. Improper removing of the cones may cause a hole in the bottom of the well and cause leakage later as you load your sample. It then may be necessary to top up the gel with more buffer to ensure that the wells are completely covered. Now we can load our gel. I will be using a 2-log DNA molecular weight ladder. This will allow me to later estimate the size of my bands of my gel. When you are loading, in this case I will load my molecular weight marker twice, one in each row.
Hello, any problem? Hello?
Yes, sir.
Sir,
There is no voice. Voice is not clear, sir.
Voice is not clear.
Yeah.
Now?
As you load your samples, be very careful not to puncture the
Yes,
bottom.
sir. Yeah. Yes. Continue, sir. Thank you.
If that is done, your sample will leak out the bottom. Also ensure that in your lab notebook you know what samples will go in what wells to avoid further confusion. Now we can start the electrophoresis. Carefully put on the lid and making sure your electrodes are hooked up properly. Remember, your DNA will move towards the positive pole. Turn on the power supply, and in this case set the constant mode, and I'll be running this gel at 130 volts. This may vary based on the type of gel, percentage of gel, and how large your gel is. It's always a good idea to make sure electrolysis is taking place, so look into the gel box and make sure bubbles are forming. And here we have electrophoresis over time lapse. You can see the dyes migrate. I like to do it until the dyes divide the gel into thirds. When electrophoresis is done, turn off the power supply, carefully remove the lid, remove the gel from the gel box, pouring off the excess buffer, and place onto a paper towel to absorb excess liquid. Now it's time to document the results. I will do this at a later video.
So now we can use these things using our tonsillimeter. We can visualize the bands. So these are all for the how we can visualize the DNA by using the gel electrophoresis techniques. So these are the basic things of gel electrophoresis. We load the DNA, then it can move faster. The smaller DNA move faster, and the bigger fragment of DNA are slower. Now another tool, that is the PCR. Now I am watching another video because it's difficult to in the small span of time to discuss the and visualize the whole PCR reaction. So I am normally I'm like to use a small video to visualize it, the PCR.
The polymerase chain reaction, or PCR, amplifies a specific DNA fragment from a complex mixture. First, the mixture is heated to separate the DNA strands. Two different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the desired fragment. After lowering the temperature, the primers hybridize to the DNA, where they bind specifically to the ends of the desired target sequence. A heat-stable DNA polymerase and nucleotide triphosphates are added. The polymerase extends the primers and synthesizes new complementary DNA strands. At the end of this first cycle, two double-stranded DNA molecules are produced that contain the target sequence. This cycle of events is repeated. The mixture is again heated to melt double-stranded DNA. The primers are hybridized, and the DNA polymerase synthesizes new complementary strands. At the end of the second cycle, 4 double-stranded DNA molecules are produced that contain the target sequence. In the third cycle, the mixture is heated, the primers are hybridized, and DNA polymerase synthesizes new complementary strands. At the end of the third cycle, 8 double-stranded DNA molecules are produced that contain the target sequence. 2 of these molecules are precisely the length of the target sequence. In future cycles, this population increases exponentially. Cycle 4, heating, hybridization, DNA synthesis. At the end of the 5th cycle, there are 22 double-stranded DNA fragments of the correct length and 10 longer ones. Cycle 6, 10, 15, 20. After 30 cycles, there are over 1 billion fragments of the correct length, but only 60 longer ones. The product, therefore, consists of essentially pure target sequence.
Now, what we already learned, that things we again visualize by a small video, that's the cloning of a gene.
First step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
So these are the other part of the, apart from our normal cloning, these are the another things that we can clone the fragmented the whole genome, and we can make the clone to making the DNA library for isolating our gene of interest. So these are all the mostly used the tools which we are using in the plant genetic engineering. Now we are entered into the plant genetic engineering. So normally here, though we are using various tools to incorporate our gene of interest in the host plant, but here I am just focusing on the Agrobacterium-mediated transformation because these are present in your graduate syllabus, especially in the BSc Botany syllabus, and that's why I am focusing only on the Agrobacterium-mediated gene transfer, not the other tools which we are using in the plant genetic engineering. So Agrobacterium is a unique bacteria, which is gram-negative, rod-separate, motile soil bacteria, and normally it has the three strains. One is the Agrobacterium tunificiens, Agrobacterium rhazogens, and Agrobacterium radiobacter. Normally, for normal plant genetic engineering, we are normally using the Agrobacterium tunificiens. These are the normally causing the crown gall disease. But for different purpose, we are also using the Agrobacterium rhazogens, especially for the hairy root preparation for the secondary metabolite production. And Agrobacterium radiobacter is a virulent strain. It's not infect the plant. So that crown gall disease is produce the this gall-like structure in the plant. And another a small video to how it infect the plants, how the naturally Agrobacterium infect the plants. So what happens if the root is injured, and normally it releases some phytochemicals, especially the phenolics, and when it's from this cut surface, when this phenolics is releases, there is a chemotactic movement of the Agrobacterium. Is Agrobacterium present nearby? They are migrated from the lower concentration to higher concentration of these phenolics, and ultimately they reach the wound surface. And when they reach the wound surface, the Agrobacterium normally a piece of their plasmid DNA they are sending to the host cell or plant cell, and that incorporate in the plant genome, and that small fragments are forced to cell to divide and redivide, and forming a gall-like structure, and these gall structure are secreting some phytochemicals which is opines, and that opines is the source of carbon and nitrogen for the bacteria, and they are getting these things as a food. So these are the unique mechanism for the Agrobacterium. Normally most of the pathogens are enter into the host cell, but Agrobacterium is a unique bacterium which is not enter into the host cell. They are present outside the host cell. They are just sending a small piece of their plasmid DNA, and that DNA having a tremendous power to divide and redivide the cell, making a gall-like structure, and they are forced to secrete the opines for their foods. So from the outside they control the plants cell, and they utilize their food from presenting in outside. So these are the very unique bacteria, and the mechanism is also unique, which is not common for the other pathogens. So normally we are utilize we are understanding gradually the molecular mechanism behind it, and we are using it as a vehicle for our own purpose. So we are discussing is one by one how we can how we are using these things. So uniqueness of this agrobacterium is a Agrobacterium plasmid. The Agrobacterium plasmid is called the TI plasmid. For T for tumor, I for inducing. That from that we are normally taking that name TI, and which are producing the hairy root. From that root inducing, that is called RI plasmid. So the bacteria which is causing the crown gall disease, their plasmid name is TI plasmid, and the bacteria which having the hairy root disease, causing the hairy root disease, the agrobacterium, they are containing the RI plasmid. And a part of the DNA which is called tDNA, which is transfer from the bacteria to the plant genome or host plant for making it a gall disease. So noted that plasmid is 200 kilobase per inside. So this plasmid is very difficult to use as a transformation vector because the high genome size is high. So these are the Agrobacterium TI plasmid. It having the virulence genes, origin of replication, opine cataplasm genes. Then there is right border, left border, and oncogenes are there, and opine synthesis gene are there. Oncogene means auxin production gene and cytokinin production gene. So normally we are these are the wild type Agrobacterium, and later on we discussed that these oncogenes and opine genes, we are normally trim it out when we are using the vector. And we incorporate our gene of interest here. And what are the uniqueness of that TDNA region? Anything if you place in between the hello, any problem with voice?
No, no, sir. But some participants muted.
Oh, okay. So what are the molecular biology behind the tDNA transfer and integration into the host genome? There are normally we have the 6 steps to integration of the plant of the tDNA into the plant genome. First signal recognized by the bacteria, which I already saw in the video. That's when plant having any wound, it release the phenolics, and that is the signal recognized by the which is recognized by the bacteria.
कितनी देर में देख लोगे? रुक जाओ तो मैं फिर 10 मिनट जा रहा हूँ कहीं. ये वाला इसे देखना.
Please mute yourself. Then thing is attachment of the plant cell. So
स्टेबलाइजर में
by the movement.
Sir, please mute themselves. Mute themselves, sir.
Kuldeep Kumar, please mute yourself. Then the after signal recognition by the Agrobacterium, it attached the plant cell, and then the weed genes, which is the virulent genes, they are expressed, and when it is expressed, the Agrobacterium produce the tDNA strand, and that replicate the tDNA strand, and that tDNA strand transferred from the Agrobacterium to the host cell, and then it is integrated into the host cell. The whole process is little bit a complex one. It is not incorporated in your botany syllabus. First, just overview. I am giving you this small pictorial diagram so that you can just visualize the things. So these are the some signal recognition protein is present. It's CHA weed A, which is recognized when there is a wound size. There is a phenolics is leaches. And that phenolics is come to the Agrobacterium surface protein, which is a signal protein, which is recognized that signal, and that signal is transmitted to the VG, and then the VG is transmitted the whole signal to their plasmid, Agrobacterium TI plasmid, and when that signal is recognized by the TI plasmid, it is replicated the tDNA, and other it also induce the other wheat genes, which is help to that tDNA to transfer it from Agrobacterium to host plant cell, and for transferring this they should make a channel. So that is a very complicated channel making by the mainly the wheat B and wheat proteins, and by this channel they are transfer the tDNA from here to here. Now that means from the host plant cell, and finally they are integrated it into the plant genome. So these are the overall mechanism of the whole things. So one thing we must note that normally from that TI plasmid, the tDNA are transferred from Agrobacterium to the host plant cell. So if we are incorporate our gene of interest inside the tDNA, it can also be transferred from the Agrobacterium to the plant cell. So by this mechanism, when we are all understand the molecular mechanism, whole molecular mechanism behind it, then we can train the whole Agrobacterium TI plasmid by our own desiredness, and for that a details molecular mechanism and molecular technology are there. So but basic thing is we are incorporating, we are normally detecting all the undesirable part of the TI plasmid and make it very small and comfortable for using as a vector, and then we are incorporating our gene of interest inside the tDNA to use as a vehicle, and that tDNA are naturally transfer our gene of interest to the plant cell, that plant which is the host cell, or they can transfer our gene of interest to our desirable plants. So these are the overall molecular mechanism behind the Agrobacterium-mediated gene transfer. So if you were using the protocol in the lab, the lab protocol is very simple one. It needs just 2 to 3 day training to using the whole things. If you have the little bit of tissue culture knowledge, then it's take 2 to 3 days training to make you comfortable for Agrobacterium-mediated gene transfer. So I just visualize it by first a small picture, and then I can visualize the whole things by the actual picture which we are collecting from our own lab experiment. So normally what we are doing, suppose we are transfer a gene to this plant. So this is the mother plant. So from that we are taking a small leaf disc, cutting it from, and then we can incubate it with the Agrobacterium for a co-cultivation, and then during this co-cultivation Agrobacterium attached. Obviously it's a genetically engineered Agrobacterium where our gene of interest is there, and gene of interest contain the obviously some antibody resistant genes for selection, reporter gene, and as well as our gene of interest. So during co-cultivation with the Agrobacterium and the plant, that cut surface Agrobacterium attached in the cut surface area, and they are transfer our that tDNA from some of the host cell, some of the cells which are adhered to the Agrobacterium. Then when we place this next step, when you place this leaf disc to the selective media, then what happened? Only the transform cells are able to survive, and non-transform cells are unable to survive. And that's why only the transform cells which are getting the tDNA, which also contain your antibody resistant marker or a selectable marker. So they select able to by this way we can select the transform cell and eliminate the non-transform cell. So these are the transform cell, and they are only able to go. Other non-transform cells are unable to grow here. And from this transform cell, if you just using some hormonal combinational change like which we discussed the earlier day, that if you increase the oxygen cytokinin concentration, that it can give rise to multiple shoots. So these multiple shoots give here, if you just increase the cytokinin concentration, or if you have the standardized protocol, by this protocol you can give rise your multiple shoot, and from that multiple shoot you can take the multiple shoot. Obviously these are the transgenic one, and from that multiple shoot you can develop your transgenic plant. And finally you can grow the plant after hardening. You can grow this transgenic plant in the outside, and now then you can taste it for whether your gene of interest is expressed or not. So these are the very common protocol for Agrobacterium-mediated transformation. So now I can visualize it, how we can do it in the lab. So these are our mother plant, where we can like to transfer our gene of interest. So we take a leaf, then cut it with that normal cut borer, where we can use the scalpel also, scalpel or forceps also, to make it a cut surface in all round. Then we can having a agrobacterium which contain our gene of interest and other things, and that agrobacterium then we can co-cultivate with this our X plant for 5 to 20 minutes, and then place it into the a media without we place it in the media without antibiotic to grow the agrobacterium as well as the plant cell for overnight. And in these 24 hours, the agrobacterium attached with the cell, and they can transfer their gene of interest, the tDNA, into the sum of the host cell. And then when we place it into the selectable media, then after selectable media, after a few days, sometimes it takes 1 week, or sometimes it takes some more time to grow your transform cell and eliminate your non-transform cell. So here's the small callus are developed, which are transform cell, and all the non-transform cells are eliminated. And then we cut the transform cell, small piece of transform cell, and place this for the formation of the multiple shooting. And then from that multiple shooting, we are placed that multiple shooting for grow, and then we can rooting and make the complete transgenic plant. So these are the putative transgenic, and now we can check whether our gene of interest are expressed in that transgenic or not. And after some other experiment, confirmative experiment, we can call it our gene of interest are incorporated here and expressed this properly. So these are the very basic things, and that's why I called it the tissue culture is the basic baseline for the production of a transgenic. And for the if you consider the botany student perspective of view, so you can contribute a lot in that part, because transgenic transgenic technology is divided into two parts: isolating the genes, identifying the genes, and design the experimental cassette. That are the core RDNA technology and molecular biological work to making your expression cassette. But when they're making the expression cassette transform into agrobacterium, then there is a whole things. Now the whole success is your hand. That's a good tissue culturist or botanist, botany student. We have a good training of tissue culture and agrobacterium gene transfer, the whole protocol. Then they can make a success to effectively to making this agrobacterium transform these things and make a fruitful transgenic. So in this part, that's why I'm last day I'm discussing that whole tissue culture, and today I'm mainly focusing on that part, so that you can interested in that part, because the background which botany students have, they are, I feel they are much more successful to be a good tissue culture as well as the plant transformation specialist, rather than the other background students who are coming from the your molecular biology or biochemistry background in that particular field, they can they have the basic all the information regarding your transformation, that RDNA technology and other thing, or even in biotechnology, they are also a good information about the RDNA technology and this part. But this part you are much more doing better. So now we are go some discuss some example of the transgenic which we are now using in our modern day time. First one is the herbicide resistance. You may know these things, and that are also included in the your syllabus in BSc botany syllabus. That's why I'm discussing some of them. One is the herbicide resistance. So if you were able to incorporate, it's a single gene. If you incorporate this single gene of interest, which is a herbicide resistant gene, in your desirable crop plants, then if you spray the herbicides, then your desirable crop plants are unable to all the undesirable weeds are unable to grow. They are killed. Only your desirable plants are able to grow. So these are the transgenic line, and these are the these are the earlier phase. These are the two after spraying the herbicides, all the weeds are died, and these are the transgenic plant. These are the before and after of herbicide spraying. So these are the one of the good example of the your plant genetic engineering product, which are also available nowadays in the market, and people are normally use it in a large scale. Then second thing and most interesting, that's the insect resistance transgenic. That's the BT plant. So these are the one of the wild type peanut plant which is infected, or normally the insect larvae are the vigorous eater. So they are eating the normally the leaves. They are highly they are getting the leaves as their foods. So normally the plants are losing their leaves. So we are reduce the crop drastically due to the insect larvae attack. So so these are the transgenic plant where the larvae unable to grow because when they're taking the leaves, they are unable to survive. So now I am giving you the how interesting it is and how it is. So Bacillus thuringiensis is also a soil bacteria. They are normally have a natural tendency when it's crystallized, when it's having a stress condition, it form a crystal, and which is toxic in nature, and that is known from very earlier time. So people are normally use this Bacillus thuringiensis as a natural insect resistant. But when that transgenic and all the RDNA technological tools are come into front line, then people feel if they able to express this Bacillus thuringiensis gene, express these Bacillus BT genes in the plant's leaf itself, then there is no need to spray or cultivate the bacteria and isolate the toxin. So it's not a very cost effective. Obviously, all the pesticide and biopesticides are not very cheaper one. It's a costly one. That's why people are using the chemical pesticides. So if we our crop plant itself develop in their body that pesticides, and if it is not harmful for us, then we can use this plant use this technology for our crop plants to prevent from the insect or insect larvae. So that's the basic things, and by which with this concept that BT plants, transgenic plants are developed. So what are the molecular mechanism behind it? When the insects are taking the leaf, that time if the plants also express the BT toxin, they also taking the protein, that crystal protein they are also taking. And then what happened when they are taking this BT protein, their insect larvae, the gut cell having a specific pH, which is in this pH the crystal is solubilized and cleaved. and it bind its effective domain is open up, and it's bind with the brass border cell of the intestine of the larvae. And when it's bind, it making a pore in the brass border cell and making the hole and kill the whole cell and whole system. And that's why they are unable to feed, are unable to absorb the food material. So due to starvation, they are killed within the 24 hours. And this crystal protein is a very specific protein which can affect only a group of insect larvae, not all the insect larvae, which are mainly detrimental for our crop plants. And that's why Bt toxin is nowadays very effective tools for making a insect resistant herbicides, insect resistant plants. So that's all for the Bt biology. And now some interesting things. So because Bt toxin and herbicide resistant, these are all incorporate a single gene into the host plant or crop plant. And now there is another global problem, and this problem is mainly in Africa and India and these are our developed and underdeveloped countries, that the deficiency of vitamin A. And a group of scientists are working with that. If we are able to incorporate or express the vitamin A inside our staple food like rice, then we can solve a problem, which is the vitamin A deficiency problem, that the night blindness problem. So for that, we should express the beta carotene inside our staple foods. And when we able to express the beta carotene inside our staple food, then when we can consume the beta carotene, our own digestive system converted it into the vitamin A from the beta carotene. So what are the basic principle behind it? The geranyl pyrophosphate, GGPP, is the common precursor of many of the biosynthetic pathways. So it's available in all the plants. So, but if you able to channelize this biosynthetic pathway, that the geranyl pyrophosphate to phytone, phytone to lycopene, and lycopene to beta carotene. So these are the only 3 steps. So all the steps involved in a single enzyme. So if we able to incorporate these three enzymes which convert the geranyl pyrophosphate to phytone, phytone to lycopene, and lycopene to beta carotene, so then we can use the plant's own geranyl pyrophosphate and we can synthesize the beta carotene. With this target, people are use the genetic engineering techniques, and this is the first time people are used to metabolize. They are incorporate changing a whole metabolic pathway. It is also called the metabolic engineering. So because they are changed the metabolic pathway of a plant and express the beta carotene, and by this way they are expressing the beta carotene in the rice endosperm and produce 21st century's nature biotechnology marvel, that is the golden rice. And now it is also available in the market. That's the golden rice. It's golden rice. Why it is called? Because it's the golden when it is expressed beta carotene. Hello. When it is expressed the beta carotene, it's gold. Its yellow is gold in color. When we boil the rice, it's look like a golden. So not only we are using to develop the beta carotene, some plant scientists are also develop the beta carotene in these maize, which are also a natural breeder. From the natural breeding source, they are also incorporate the beta carotene in. So not only the genetic engineering pathway, people are also enrich some of our staple food by the natural breeding also. But they are lucky that whole biosynthetic pathway are present in their common ancestors, so they are able to incorporate it. And we also making some cauliflower also, which are enriched in beta carotene. So these are some good things of which are already available in the market. And the next problem is the water crisis. We don't know the most of the water are used, most of the sweet water are used for the agriculture purpose. More than 70% of water which are sweet water which we use, that are used for the agriculture purpose, and few water are used for the industrial and our household purpose. 70% water is used for agriculture. So obviously we all know we have a sweet water crisis in near future. So obviously we should look into the matter, especially the plant biotechnologist or the plant scientists are look into the matter, so that we should reduce this consumption of the sweet water for the agricultural purpose, or else there is a huge crisis in future. So and obviously the coming drought are also a problem for globally to reduce the inter crop production. And we are lucky then some companies are working on it, and they are already commercially developed some varieties which are need a very less amount of water. I'm not going in detail, but I'm giving the information so that you are getting interest in that information. If you wish, you just type in Google, you collect all the information regarding this. Then we are discussing some concern about the GM activities. So normally what happened, we are incorporating our gene of interest, and that gene of interest expressed in our host plant, and that genes are expressed in everywhere, in the root, in the fruits, in the leaves, other. So in primary stage, people are using the genes, they are using the constitutive promoter so that the genes are expressed everywhere. But now the technology are developed, so now we have the tissue specific expression. So if you wish, we can express our gene of interest in a very specific tissue, not the all the cells, all the part of the body. We can also having an inducible promoter. So by using the inducible promoter, we can control the expression of gene also, that when required we are expressing the gene, and when not required we can stop the expression of the gene. So not only in the tissue, but we also regulate the expression of the gene of that particular tissue itself, that whether it is expressed or whether it is not expressed. So these technologies are nowadays available. So that are the some more interesting things you should know, which are not in your that undergraduate syllabus, but later on you gradually understand this if you are interested. Then we are also giving you some other information, which are the some people which are against to incorporate the gene of interest from the other sources in the plant. They are also incorporate the cysteine. What is cysteine? We all know in genetics there is a two term. One is the cis, another is trans. So we obviously call it transgenic because it's coming from the other sources or transform. But we can also use these genes for as a cis transfer. So from the same type of genes, from the same type of plant, if you transfer the gene, like from one variety apple contains some good genes and other variety does not, we can transfer this gene from the these things to this red apple to green apple. So obviously it's called a cis transfer. So in that case, people are doing these things. So these are the another new terminology, that's cis genetics. Now another things also we are doing by the plant genetic engineering, that silencing a gene. Sometime if you just blocking some genes which is undesirable, then you can also getting some good characters. Like a very good example which is already available now in the market, that when we cut the apple, we are getting after few minutes we are getting a brownish color in the apple. So this due to a presence of phenolics in the high amount of phenolics in the apple, because these phenols are oxidized and form the browning color, which is in a high amount, which is also toxic in nature. So people are just just block the expression of certain genes, which stop the overexpression of the phenolics in the apple fruits. So that also solve this type of problem, which is agriculturally or a commercially beneficial trait. So we can, if you just stop the expression of some genes, then you can getting some commercially beneficial traits incorporate in your fruits or vegetables in other platform. So the plant genetic engineering also help not only the introduce the gene. What I like to say, because most of students having the concept that transgenic plant means it only express a transgene from the other source. It may transfer the genes from the same type of plant source, and not only that, it can also block a expression of a undesirable gene in the plant through this transgenic technology or through this this plant genetic engineering. We can also suppress the expression of some gene also which is desirable. So not only the incorporate the other genes, but also the stop the expression of the same genes, their own genes. We can use this transgenic technology. Okay. Now these are some very recent advanced features of this whole plant genetic engineering technology. One is that we can make a transgenic grafting. So these are the lower part is transgenic and upper part is non-transgenic. So if the desirable part is the lower part, that means if your specific trait is a pathogen resistance which is normally affected the root, so you can make a transgenic root part and grafting the shoot part, which is the natural normal one. So your pollen and other thing is upper part is non-transgenic. So you just use this plant as a grafted plant, and which the lower part only the transgenic and upper part is non-transgenic. So that can be done. Another is the new another things. If the lower part, which is transgenic part, if they are developed some, they are express some RNAi, and which is ultimately block some of the gene of your interest. So that can be also developed in the lower part, and upper part is the normal, because RNAi, now we already know the RNAi can migrate through our vascular system, and they can act in the upper part also. So by this way, making a chimeric plant or grafting plant, we can also develop the transgenic. These are the very new approach of transgenic, that plant genetic engineering, that are also used by the different people. Now the other very recent things, that we can also precisely mutate a particular gene of interest or knock down a particular gene of interest or incorporate a small sequence in it by this zinc finger nuclease technology, or the TALENS technology, or very recently maybe you heard this term, that's CRISPR-Cas9 system. We can also use the CRISPR-Cas9 system in the plant genetic engineering for blocking a specific gene, or making a very specific mutation, or incorporate a very small fragment in our gene of interest, or corrected the mutation. So by this way we can do all these things. Now these tools are available, and these are the very recent tools. If you are interested, because these tools are much more complicated one, it's very difficult to understand these things in a very small period of time. So I just giving you these ideas, so that or terminology in your mind, so that you have if you have getting interest in that particular topics, you can search and you can read more about this. Or if you have if you face any problem, you can also mail me. I can give you some other videos and other relevant articles, so that you can self read yourself. So, Now some future challenge.
Novel microbial formula.
So this future challenge, we have the problems and also having the solution. So we should improve our nutrition content. We should improve the stress tolerance. We should increase the disease resistance. We should increase the water use efficiency of the plant, higher yield, obviously, improve the fertilizer uptake by the plant. So these are the very basic needs which is asked for the different plant biotechnologists to develop to do so. So these are the not only the all the areas. I'm just giving ideas of some of areas which are the now a very pioneer areas for the plant genetic engineering. Now, obviously, we are all are waiting for our second green revolution and for less damage of our environment and giving the more food and more other relevant plant product to all the people who need it. Now, what we discussed today, what is requesting by the organizer to discuss some of the part of the for the basically for the BSc Botany student for their career perspective view. Especially, I focus on first. I like to focus on it how from the science they are shifted to the technological field. So maybe some people are interested to this type of work or this type of research. So I hope most of the students or most of the viewer are the BSc Botany students. They can do their own MSc in Botany. Then they can appear for the GATE Life Science or GATE for Biotechnology. I feel GATE Life Science is very comfortable for them. So they can clear the GATE Life Science and then they can enter into the MTech Biotechnology or MTech Agricultural Biotechnology. Some of the like IIT Kharagpur also offer the agricultural biotechnology that are much more better for the, I feel, the MSc Botany student. Or they can opt for the MTech Biotechnology also, because agricultural biotechnology is not offered in so many institutes. So they can enter into the MTech Biotechnology program. And then they, after MTech Biotechnology, they can enter in any industry, not only the plant industry, but also the, if they are in the biotechnology and MTech, so they have a vast horizon of area. So they cannot confine with the plant science, but also they are also opt for the other industries also. At the same time, they can also hired by the industry for the research scientist. So other ways they can, if they are very much interested in technology, then they can shift themselves from the BSc Botany or BSc to the BTech after completing their graduation. If they have the mathematics in their +2 level, then they can enter as a lateral entry for the BTech Biotechnology course in second year, and they can complete their BTech course also, and after BTech they can do MTech and other things. So these are the two ways of making the opportunity to those who are in the science stream. They can enter into the technological stream or applied stream. Because I feel after this pandemic, the whole applied biotechnological field, especially the pharmaceutical sector or pharmaceutical biotechnology sectors, are now upcoming, and we all realize what is the potentiality of the of these type of research or these type of activities for all of our our stakeholders. So these are the few things. If you have any queries, you can write in the chat box, or you can in the message box, or you can interact. If you have any query, you can also interact. So that's all. Thank you. And there is a email ID. This is my email ID. You can mail me if you wish. So now it is I'm hand over it to Arjun. Hello. Hello.
Sir, this is Dr. Ovik Sabanto. I think
Huh.
Arjun is not there. So
Huh.
we are very much thankful to you that it was indeed a very elaborate lecture, and from the very basic thing you have gone through mm, the deeper into the topic. So the student as well as the participant which are from different backgrounds, some of are from the faculty members, some of are in the MSc and doing research. So all of the participants have enlightened by you and on the field of biotechnology, and you made a connection between botany and biotechnology. Now I would request the participant to ask their queries or question either on the chat box, and after that we can go to the live interaction session. So initially we have some question that the participant have asked in their chat box. Then after, if the time permit, then I will go to the live interaction session.
Hmm.
So there are some question that Subhadeep they have asked that when plasmid inserted in, all it is there any anti-reaction by the cell that because as we know that the cell is not initially permit the plasmid, so it is there any anti-reaction by the cell during the insertion of the plasmid?
No, we are fortunate the bacteria won't have any antibody. Okay, and normally for survival of a plasmid inside a bacterial host, we have to think mainly the two things: whether the origin of replication is there, that means that origin of replication is compatible with the host, so that the plasmid can replicate. The second thing is obviously another thing is restriction endonuclease. So that normally they also having their own methylase system, so they are not interact with the plasmid. But if the original replication is fine, they can replicate inside the host. So like the animal system, they won't have the immune system in their body. The bacteria won't have the immune system in their body. So normally it's not create any problem.
Thank you, sir. Another question that he
And
has
another
asked
thing
that
I'm incorporating this
yeah.
thing, that host we are using normally the E. coli or other bacteria for cloning the plasmid or expressing the things, that bacteria also their chromosomal they are also is a genetically engineered bacteria, so that that problem should not create. Okay.
Thank you, sir. Thank you very much. The another question is there that can we stop potato rotting then normal time using this biotechnology? Because
Yes,
I think
yes.
this participant is from
Yes,
question.
yes, already we have this technology, and if they search it already, we have the published paper, and in that particular field, that are people are working that and successfully develop the technology. So that is the another good field of research. Obviously, they are getting if they search, they're getting some papers. We can stop the potato rotting by the fungus attack.
Okay, sir.
Hmm.
Okay. So another question is from Arubraton Das. Dr. Arubraton Das has asked that is there any crop or vegetable that are genetically modified available in India, which we normally take in daily basis? Either there are some crop or food is there available?
Uh, some crops are already commercially available in India. I feel if you nowadays if you go any supermarket like our Big Bazaar or your Spencer shop, you can get the golden rice. Okay, obviously it's not a very cheaper one. That's the another controversial part, because golden rice at present is not commercially cultivated in India. It need another 2 to 3 days for getting the permission of ministry. but it already cultivated in the other part of the world. So now it is available in the and that's why cost is little bit high. It's already available in the supermarket. And after BT cotton massive controversy in South India, government having a very strict regulation, and that's why it's delayed in our country for commercially available crops of transgenic crops. But within 2 to 3 years, we are getting a lot of transgenic crop in Indian market.
Okay, sir. Sir, another question is from Ananya Dwivedi. She asked that can whenever we are doing the tissue culture, then we initiate the root first, then shooting in vitro culture. Is it possible, or always there is possibility of only root or only shoot?
Now, in tissue culture, if you wish, you can develop the roots. It's not a problem. We can, if you just making initial callousing, and then if you increase the oxygen concentration, you getting a profuse root. Okay, but what happened normally? The shoot is normally that the conventional way. If you are not interested for the root, one thing is if you interested for the roots, then you can develop lots of roots. It's not a problem. But if you interest for the entire plant, then first thing is multiply this plant. And that's why we are using the multiple, that there is a step that is called multiplication. So if you n number of multiple shoot, then you have the n number of plantlets, and from that n number of plantlets, you can develop the roots. So you can getting a individual plants. And that's why the conventional way is making the multiplication, then shoot elongation, then rooting. That's the conventional way. But if you interested for the root only, if you like to isolate something which is present in the root, you just develop the root only. Even in hairy root culture, we can just culture the root only. In the liquid system, we can develop the profuse root. Not any it's not like that. They only, if you develop the shoot, then only we can develop the root. It's not like that. It depends on our desire or which we need.
Thank you, sir. Another question is from Abdul Shakil Khan. He has asked that can two different genes can be inserted in the same plant by using two type of Agrobacterium strain? Is it possible, or there any
two types of
way? He's asking that two type, if we want to transfer two type of gene on a single type of in a single type of plant, I think
normally we are using more than one gene to incorporate into the using a single cassette, because in a single cassette we can incorporate more than one gene. It's not a problem at all. And if you just consider the golden rice technology, there the number of gene is more, number of gene is 3. We should incorporate 3 gene to complete the whole biosynthetic pathway, and then the tDNA size is enlarged. It is difficult to transfer the whole tDNA into the host cell. So in that case, we are using 2 agrobacterium. In one agrobacterium, we incorporate 2 genes, and another agrobacterium we incorporate 1 gene, as well as in that agrobacterium we also incorporate your reporter gene and selectable marker. And by this way, we are using 2 different agrobacterium to incorporate the more than 3 genes. Okay, so we can use the 2 different agrobacterium to incorporate more than 1 genes if it is needed. And it already used in the golden rice technology when we develop the golden rice. Okay.
Okay, sir. Sir, another question is from Choitali Das. She has asked that though the eukaryotic genome is very large, so how the restriction enzyme recognize the palindromic sequence in the large fragment of the DNA?
Restriction enzyme is recognize the sequence. It's not depend on the size of the genome, and that's why when you use the plant genome or animal genome or any eukaryotic genome, as it is very large, so if you use a single restriction enzyme, it cuts the restriction in the whole genome, in a multiple of copies, multiple of different size of fragment, and which is we can visualize by the gel electrophoresis, because number of cut size is less and number of fragment is also less. But if the plant genome is huge, then it restriction enzyme it cut very frequently, and if you run the in a gel, you are not able to visualize any fragment, you are getting a smear, because if a simple statistics if you are using as the number of base pair is 4, that ATGC, and restriction enzyme recognition sequence if it is the 6, then the probability of cutting of that restriction enzyme in the whole genome, it is it is the 6 to the power 4 to the power 6. So it's round about 300 3000 kilobase kbs. That means only after each and every 3000 kbs you are getting a cut. So you are it's a because normally it's a 150 to 200 MB size of genome. So you are getting a huge number of cut, and you are getting a smear only.
Okay, sir. Another question that asked by Subhadeep Dey, that is either BT cotton is in some recent news that it has had negative impact. So either it is applicable to Indian context or not. Is it? It could be applicable or not?
BT cotton is not negative. Any negative negative impact is what happened in the South India due to the high temperature. Somehow that BT cottons, BT genes are not work properly. So when the farmers are using the BT cotton, they are not using any pesticide. But if the protein is not folded properly, so what happened? They are not effective at all for the insect, and that's why a huge damage caused of thousands of acres lands which are used the BT cotton. They are damaged by the insect itself, and that's why a huge crop loss takes place. But there is no report that BT cotton having any toxic effect in the human consumption. So it is not this point of view. It's not a harmful one. But there is a controversy that we need some time for human trial, and that's why the BT brinjal when come up in India and Monsanto is try to introduce it, there is some protest, and people are deferred these things. In government of India deferred these things. That obviously we need some more time to getting the things approved by the different agencies, that whether it have any toxic effect or not. But till now there is no such report of toxicity of the BT gene in human consumption or cattle, even in cattle. And that's why I say that some other technologies are also used for transgenic purpose. Not only the transgene, we can use the cisgenes for transgenic technology. We are also making the gene silencing or suppress express of undesirable gene by this technology. And we can get some new traits or desirable traits for that. And that's why I'm in last few slides I also expressed, I also covered up some new ideas and new technologies which are available to overcome this type of controversy.
Okay, sir. I think this is the last question in the chat box that someone has asked, that is either any animal gene can be transferred through the bacteria to the plant gene, or it can be expressed.
Obviously, animal gene can express in the and there are some there are so many commercial plants are available which express the animal genes. Like last day I expressed that we can develop the vaccine inside our fruit plant, like banana, then papaya. We can express our vaccines, as well as I can give some pharmaceutical examples, like like there is a protein is called antiheparin protein, which is isolated from the leaches, the hirudin. Hirudin is leaches. That protein name is hirudin, which is an anticoagulant protein, which is expressed in the which is useful for the stopping the clotting of blood. And this hirudin protein is used for the basically for the heart patient regularly. And now this hirudin is overexpressed in the, because there is a hirudin is a very small protein. So if you express in the bacteria or in bacteria, then it is difficult to fold it. And if you express in the yeast system, then it is very difficult to isolate it from the yeast system. So people are incorporating the plant system in a very wise manner, and now it is expressed in the brassica seeds, and people are harvesting that hirudin commercially from that brassica seeds, and it is available in now we are using that commercially in the pharmaceutical industry. And by this way we can contribute a lot for the pharmaceutical industries for expressing their small molecule in the plant system. I just giving a very small example, like we are using the spinach. And spinach is a leafy vegetable, and we all know now the chloroplast engineering technology, we can express the gene, our transgene, in a very huge quantity if you express it in the chloroplast, because number of chloroplast in cell is more or less 100 to 200 per cambium cell. So in the palisade parenchyma cell in the leaves. So obviously if you incorporate it in the chloroplast, the number of copy is much more higher. The number of copy is 100 to 200. So we can, if we express a gene in the chloroplast, and if it's the biomolecule, small biomolecule, there is a lot of advantage of that, and we can express it in a huge quantity. And if you use this expressing system in the leafy vegetable, then we can take the whole plant. We can cultivate it throughout the year with the control environment. We can cultivate, just we take the leaf and we just extract the biomolecule. So only things, if you develop the transgenic, then you can there is no need of sophisticated fermenter or other things for running the industry. You just having a downstream processing unit with your hand. You just give the seeds to the transgenic seed to the farmer, they are cultivated, and if you pay them double, they are not using this product for the other purpose. They are obviously sold to you for that particular industry, and they can harvest the things for isolating the biomolecule, and it's very much cost effective. And nowadays the most of the pharmaceutical company invest money for the to the plant biotechnologies to develop this type of transgenic, and that are the another potential area for the plant biotechnology research or plant scientist to express the biomolecule for the pharmaceutical industry.
Thank you very much, sir. So we have very less time to go to end the session. So if there are any question that student ask directly to our sir, so they can raise their hand and ask their question. Initially they can raise in the chat box and ask the question. We'll wait for some time, then after we'll end the session. Yeah. Other anyone have your question? You can ask.
I like to interact any of your student
Yeah.
from your two colleges, especially for whom we are like to you are arranging this seminar series. If any of them,
yeah. Yes, any student can ask your question. just unmute yourself and ask your question.
Don't
Then
worry,
Sir is
you
there
can ask in
to
Bengali
answer.
also. Don't worry, you can ask in your question in in Bengali also. Don't hesitate.
Hello, sir.
Huh?
Sir, I'm Kuhili Vomik, and I am from plant science background. I have done my BSc and MSc in botany, and in this year, in 2020, I have qualified GATE exam, and All India rank is 538. So, what can I do now? Either I am go for PhD or either I am go for MTech. But in my MSc, my special paper was cytogenetics and plant biotechnology. So, sir, your session was very interesting and very informative. And I was doing a project on genetic tissue culture, plant tissue culture on uh, peanut or Arachis hypothia. So I am now confused. So whether I am going for PhD or whether I am going for MTech. So, sir, please give me any suggestion.
It is you should think about your future. What do you like to do? If you are like to enter into the academic field, especially in the teaching, so in that case you should have a PhD degree, because nowadays what are the UGC regulation, you should have the NET as well as your PhD degree for enter into the college or university academics.
Yes, yes, sir.
Hope Ovik, is it true?
Yes, yes. Now the PhD is mandatory for
PhD
college
is mandatory,
certificate.
and as well as the NET is also mandatory.
NET
But sir,
new.
Huh?
Yes, sir.
But sir,
Continue.
I have little bit interest about biotechnology related industry
So
job.
no, exactly. That's why I'm saying for because the PhD it's a more or less 3 to 5 year venture.
5 years, right.
3 to 5 year venture, and during this PhD time, you should have cleared the NET also.
Yes.
To ensure your job in the academics. But MTech is the 2 year venture. So most of them now, when you complete your MSc, you are not very much grown enough. So you can spend 2 years for MTech, and you are getting much more advanced tools, and also having a good opportunity to enter into the industry. So after 2 years, if you feel you like to enter into the PhD, then you can enter into the PhD. That line is open for you. And if you like to enter into the industry, that also, so you have, if you just spend another 2 years, then you have two different avenue. But if you enter into a PhD, you have only one avenue. That
Yes,
either
yes, sir, exactly.
in the academics only. So I hope within the now we are completing our masters within the 16, 17, 18, 19, 20, 21, 20, 23, 20, within the 23, 24. So we can spend 2 years for MTech, for getting more to open up more avenues. And when you doing your MTech project in 1 year, that time you are getting much more exposure in your particular field. So that time, if you interested in industry, you can do your project in the industry, so you can directly absorb in the industry. If you like to do the PhD, you choose your right field, which your subject of interest, and then doing the MTech work in that particular field, and then you can continue that work to the PhD. Then you can reduce your PhD time also. That means you just losing in that case in 1 year only for the academic other getting the other subjects, which are theoretical subject and practical subjects, which is taught in the MTech syllabus. So you just in that case you can spend only 1 year, losing 1 year. But when you getting the GATE scholarship, then you are getting the scholarship also when you doing the your MTech work. So with scholarship, you are allowed to study for 2 years for higher study. So why not you taking this opportunity?
Actually, sir, someone has told me that as we are from biological background, that MTech is not beneficial to us.
No, no,
Actually, is that right?
no, no, it's a pure wrong concept, because I have a lot of students who are
Yes.
MSc in botany or MSc in microbiology. They are doing their MTech in biotechnology and
Yes.
enter into the industry, in a different industry, and they are doing the good work. Even in your Vidyasagar University student, I have.
No, sir. I am from Lady Brave on Calcutta
Okay,
University.
no. I have some Vidyasagar University student in my MTech, who are
Okay.
basically in the microbiology and botany background, and
Okay.
some of them now work in the Bharat Biotech. You all know the name of Bharat Biotech, and
Okay.
I am getting that COVID, all the information, whether the vaccines they are developing or from that students only. They are sending the information.
Yes, yes.
So
Means that, sir, if I go for MTech, then there is any chance for getting a job, right, sir?
obviously you
Okay,
have,
okay.
now you have the two different avenues. You can, after MTech, you can go for PhD, as well as after MTech you can go for the industry. So you choose your own. You may after 1 year you may not feel that I am not comfortable. When you, if you interested in the industrial work for your
Sir,
MTech, there is
I have little bit interest about that industrial
no,
job.
no. You don't have the industrial exposure. Maybe
Uh-huh.
after entering into the industry, you may feel that it's not my job. Because what
Okay.
I feel that most of the students are very much excited to enter into the industry, but
Yes.
industry having a different environment. Some
Yes.
work, if you are not in the R&D, it's not obvious you are put into R&D. If you are not in the R&D, if you are in the production department, then it's a routine monotonous job, something like that. And if you are in the R&D, then obviously you also having a huge pressure, that obviously all the industry, they are try to do their work in a very time frame manner. If
Yes.
nowadays the COVID vaccine, all the government, all the sectors are forcing the industry or the researcher, those who are industrial research, that develop the vaccine within the 3 months, within the 4 months. So obviously you have a huge time pressure for each and every production, if you are in the R&D or in the production. So sometimes we are not mindset in that to cope up this pressure, because industry give you much more higher salary than the academics, but they are also giving you much more higher pressure than the academics in a very much reluctant in compared
Yes.
to industry. So in that case you are getting a exposure. If you are interested in the industry, then you can do your project 1 year dissertation project in industry. So in that 1 year, you are having an opportunity to closely monitor the industrial activities, and then you can feel you are not suitable for the industry, then you can shift it to the academics.
Okay,
Okay,
sir.
so
Sir,
that's
thank
why,
you.
that's the another opportunity for the doing the impact.
থ্যাংক ইউ সো মাচ স্যার। অ্যাকচুয়ালি কিছুজন বলছে কি মানে আমরা যেহেতু কোর বটানি ব্যাকগ্রাউন্ড বা বায়োলজি ব্যাকগ্রাউন্ড, বলছে আমাদের এমটেকটা আমাদের জন্য বেনিফিশিয়াল নয়। তো খুব পাজলড ছিলাম যে কি করবো, এদের পিএইচডি তে যাবো না এমটেকে যাবো। বাট আমার খুব ইচ্ছে যে ইন্ডাস্ট্রিতে জব করার বা এই বায়োটেকনোলজি রিলেটেড
আমি
মানে
তোমাদের
নিয়ে
বলবো,
থাকার।
তোমাদের অনেকেই আছে যারা পিওর বটানিতে এমএসসি করে তারপর ইন্ডাস্ট্রিতে খুব ভালো কাজ করছে। ইভেন in this 1 year they are develop their immunology skill, their animal cell culture skill, and they are now working in the pharmaceutical research, not in the plant biotechnology field. They are doing well in the pharmaceutical research.
Okay, sir. So thank you so much, sir.
Because in the higher study there is no barrier.
Yes, exactly,
A good chemistry student can do a very good research in biological field. A very physics students can do a very good things in the field of biology. So why not a botany student are able to good in that biological field?
ইয়েস ইয়েস স্যার। স্যার এমটেক এর জন্য কোথায় কোথায় মানে চান্স বলছো যেহেতু আমার
আচ্ছা,
গেট
I
আছে তো বলছে
think
আইআইটি তে।
I should interrupt, sir. আমি একটু interrupt করছি
হ্যাঁ
because time is running out. We have to give all the participants
yes
some
yes
chance.
yes
So
yes
আমি বলছি কুহেলি, তোমার যদি কোনো পার্সোনাল কোনো থাকে, স্যারের তো মেল আইডি আছে, স্যারের
ওকে
সঙ্গে
ওকে ওকে
ডাইরেক্ট তুমি যোগাযোগ করতে পারো, কোনো প্রবলেম নেই। আমরা তো স্যারকে বলেছি, স্যার ডাইরেক্ট মেল আইডি দিয়েছেন, সেখানে স্যারের ফোন নাম্বার
ওকে
যদি স্যার
ওকে
দিতে
ওকে
চান
ওকে
দেবেন,
থ্যাংক
তুমি
ইউ।
ডাইরেক্ট কথা বলো। But it is
ইয়েস
a
ইয়েস
seminar, so you have to
ওকে
make all this
হ্যাঁ
arrangement to all the participants. Sir,
ওকে ওকে থ্যাংক ইউ সো মাচ স্যার।
yeah, sir.
হ্যাঁ
So I think you have questioned a lot about him, about her and all the participants. এবং আমরা যদি if any participant have one or two questions, then after we will conclude this online lecture, because we have another schedule from 2 p.m. So anyone can ask question from the academic query or the subject related query. Anyone, one or two questions, then after we will conclude the session. Yeah. Yeah, I think we have you have answered. Yeah, one of anyone directly can interact with Datto, sir, Professor Datto, with his email ID, and if he agreed to give permit the phone number, you can get it from him, and after that you can interact with your educational background, because he has a vast experience on teaching BTech, MTech, and as he is from the botany background in BSc, MSc, I think it is the good. He is the good example of combining of basic science and the technological science. So that is why we we rather invite him to give a lecture, not only lecture but also for your career objective. So very thanks to the sir, and I will request if Momtaz is there, so she can conclude the session. Yeah, Momtaz.
Yes, Bhumika.
Yeah, you can
Uh,
conclude the session. Okay. Yeah, Momtaz,
Thank you, sir.
concluding.
Sir has joined with from 2 days, 14th July and 15th July, and delivered the two topic. In previous topic, why should we learn the plant? This is from root basic to upgradation, and today, sir, focus on plant tissue culture and plant genetic engineering. The genetic engineering is very effective topic and present scenario. Student cannot learn in our traditional uh, study. Previous and sometimes Kueli Bhumik is asked sir, and so he's interested in the topic. So thank you, sir. You spent your time from 2 days within us, and again, thanks for sharing your views, your thought, your experience from your study level and career also. Why take student can why they learn, why should take the part in future in their career. So thanks, sir, and uh, in future we all call you again. We organize such a program, and I think you respond positively and join with us. Thank you all participants, dignitaries, research scholars, students in our 1 week online series. And we again meet on 4 p.m. today, the next series. The next speaker, Professor Ashish Nandi, and the topic is plant immune system overview. So we again meet on our lecture series at 4 p.m. onwards. Thank you so much. Thank you to all. And we end our session here. Thank you.